This is really exciting for me because I remember when I applied to join Cold Spring Harbor as a faculty, one of the first things I did was look up some of these cocktails and chromosome videos to get a sense of what it was like. And I think I saw Lloyd Trotman give a talk during No Shave November. So he was doing like a rather shaggy cocktails and chromosomes. So hopefully I can capture that energy as well. So today I'm going to be talking about how the brain, in a sort of very vague sense or an overarching sense, communicates with the body, and how the body communicates with the brain reciprocally. So as Leah mentioned, we study cancer and how it interacts with the body. And we focus on a few different brain regions that really orchestrate this bidirectional communication. And one of those regions is the hypothalamus. So this is a slice from a mouse brain where we have different cells labeled with different colors. And this brain structure I'll talk about in a little bit, but it's one of the major ones that is responsible for integrating information from your body and then sending signals out to tell your body what to do. And something I thought would be relevant for tonight is that actually this structure, there are neurons in this structure that project all the way down to your liver, where they control metabolism of alcohol. So this part of your brain actually controls how drunk you get, basically. Okay, so before I jump into the science, I guess I'll give a slight detour as to how I came to be here in the first place. And also for the younger people in the room who are looking to potentially have a career in science, I wanted to highlight the point that no path is a straight line. I didn't do the sort of, linear path upwards into where I am now. So I'll give a brief overview of sort of where I'm coming from so you can get a sense of how I think about these things. So I grew up in DC, Washington, DC, and I graduated high school in 2007. I had no idea what I wanted to do. I just knew that I liked scuba diving. And I liked interacting with the biological world. I knew I liked biology more than finance, so to say. And so I started to think about that, and as I did, I went to Indiana University, because if you're not aware, if you're from Washington, D.C., in the city boundaries, since D.C. is not a state, you get in-state tuition at any state school, which is really nice. But we don't get a voting representative, so that's the truth. So after college, I actually got my degree in biological anthropology, and I worked with chimpanzees for a while, which was a lot of fun. But during that time, I realized I was very much interested in the biology, but I was very much reserved to only observing. There was no tinkering or manipulating that I was able to do. Obviously, for logistical and ethical reasons, we can't do that. And so I wanted to dig deeper into the circuitry in the brain, which ultimately controls behavior. And so I convinced a admissions committee that even though I didn't have a degree in neuroscience, I could get a Ph.D. in neuroscience. And so that's what I did. And then I got my degree in 2017 from Ohio State. And then I started a postdoc at Stanford, and then I came here in 2020, right before the pandemic threw everything into chaos. Okay, so that's a little bit of my background. So what I really want to get across today is three components of this dynamic. And that is, how does the brain sense things going on in your body? How does the brain interpret that information that's coming up from the body? And then how does it signal back down to the body to influence physiology and behavior? So you may have heard of the Nobel Prize that was awarded a couple of years ago to David Julius and Artem Padapushin, who were actually here a couple of months ago at the Brain Body Symposium that we had at the lab. And they won this Nobel Prize in physiology or medicine for discovering these critical receptors which drive the sense of touch and the sense of temperature and heat, or certain types of pain. So the first one is called TRPV1. It's a capsaicin receptor. It's the thing that makes chili peppers taste hot. It also senses a lot of other things. The other one is called a stretch receptor. It's called piezos, but they don't stretch or they don't sense, whole body stretching, like stretching your leg, like here. They sense cellular stretching, like if your bladder is filling up, the cells in your bladder need to sense that stretch in order to tell your brain, hey, you need to go to the bathroom now. So these are very important. They're involved in determining your blood pressure, for instance. Your blood vessels have little sensors in them that tell your brain, hey, let's bring down blood pressure a little bit. And these are part of that circuitry. And so where are these types of sensors? They are on nerves, and they're on sensory nerves, and sensory nerves sense things. And so they cover your entire body, on the inside of your body and in your skin. So that's how you feel touch, and that's how you feel, for instance, pain in your stomach if you have a stomach ache. So they detect things like heat and stretch, but they also detect many other things that are relevant for keeping us alive and keeping us healthy, such as pain, acid and CO2 levels, glucose and other metabolites. Inflammation is also sensed by these nerves, et cetera, et cetera. So there's many, many things. Virtually any biological process that's happening in an organ is being tapped into by the sensory nerve. Okay, but sensory nerves aren't the only ones that sense things. The brain actually senses things itself. It doesn't need the nerves in the body to send that signal up. It can also sense things that are arriving through the bloodstream. And I like to give this example because this is a very fun example to talk about, which is this finding from the 90s. This is a picture of two sibling mice. So these are brothers, except this one is lacking a gene called OB that codes for a hormone called leptin. Does anyone want to guess what the OB gene stands for? Obese, yes. It stands for the obese gene. And this is not just a weird thing that happens in mice. There are humans that lack leptin as well. And these are the patients here in A, B, and C. And they lack this gene. They lack the OB gene. However, if you give them low dose of leptin, they lose all of that weight. And these two people that were not labeled here, those are the nurses that took care of them. So, yeah. You can see how basically strong the signaling pathway is, and how this works is that these mice and these people don't make leptin. And what leptin does, and it's made by fat cells in the body primarily, it tells the brain through the bloodstream that you're full and that you should stop eating. So if you don't get that signal, you never get that feeling that you're full. And so this mouse will just keep on eating until basically it can't eat anymore. And so if all we have to do then is, because the receptor for this hormone is in the brain, all we have to do then is give leptin, because we already have all the machinery there for that molecule to tell the brain what's going on. It's just that we are lacking the gene that encodes that molecule. So if you just give low dose leptin, they lose all of that weight within a couple months. So it's a very powerful pathway. Okay, so what happens when that information gets to the brain? Where things are coming up from your body telling your brain, hey, you have a blockage in your GI tract, or hey, you need to metabolize this food, or hey, you need to increase your heart rate, or maybe your breathing needs to increase. All of that needs to be interpreted by the brain. For instance, if you are sleeping and you roll over on your face, how does your brain know to engage this gasping response to wake you up? All of that requires this sort of integration that happens in the brain. So this is a giant field of neuroscience, understanding how information is integrated and understood in the brain. But at the sort of base level, what is happening in every cell in the brain, in terms of basically the simplest version of learning, is that since nerves are electrically active, so if you inject electric current into the nervous system, you will get neurons to be active. And so we can use that as a way to manipulate nerves, but also monitor what they're doing. And so when nerves fire, maybe you've heard of something called an action potential, for instance. Action potentials are an all or nothing signal. So what happens is that inputs from a bunch of cells come into your brain. So there's, say, information on your heart rate, information on your breathing, information on your blood pressure, all coming into your brain. And certain cells need to interpret that signal. And the way they do that is through integration, where an input comes in, maybe another input comes in. So say like hunger, thirst comes in. But if they're far enough apart, or they're not really, like you're not very hungry, you're not very thirsty, they don't stimulate the neurons because they don't summate. But if you have two inputs that come really close together, like say pain and heat, then certain neurons will add those signals up and fire a big action potential. And that is fundamentally what's happening when the cells in your brain are interpreting what's going on. So that was a little bit about how do we sense signals coming up, how do we interpret signals that reach the brain, but let's go the other way around. What happens when we turn on certain populations of cells in the brain to the body? And so I'd like to give this example. So this is a very tiny nucleus in our brains called the locus coeruleus, or the blue spot. It literally looks blue in human brains. So this is a human brain slice. It's these tiny groups of cells here, and they make the majority of a molecule called norepinephrine, or noradrenaline if you're from overseas. And so what we can do is we can manipulate these neurons using a really cool technique that's been in neuroscience now for about a decade, a little bit over a decade. And that is to control these cells with light in a technique called optogenetics. And as I mentioned, these neurons are electrically active, so we can just turn on neurons with light very easily. And let me go back a little bit so that you can see this video from the start. Okay, so essentially what I wanted to highlight here is that the nervous system, if you stimulate these neurons, these tiny group of cells in the brain, you can elicit whole body norepinephrine release. And what is norepinephrine? It's a very important molecule that signals information from your brain to your body. And as an example of why this is important, this is live imaging in a lymph node of a mouse. And these are all immune cells running around. And you can see when we pulse norepinephrine, they all freeze. So you can imagine how powerful that signal is coming from the brain, causing the entire immune system to freeze. You imagine that these frozen immune cells would be pretty bad at eliminating a pathogen or fighting cancer or repairing tissue, for instance. And so we're very much interested in how information from the brain elicits downstream processes in the body that will influence disease. And I'll give another example of how important these specific neurons are. These are the neurons that really control what's called a fight or flight response. And you'll see here in 1 second, when the video hits the midpoint, I'll explain in 1 second. We turn on a laser and watch the pupil on the mouse. So this mouse is anesthetized. It's in a little nose cone, so it's asleep. And we have a fiber optic going into its brain that you can't see. And it's right sitting on top of those locus coeruleus neurons. And what those cells really powerfully control is our fight or flight response. Many of you have probably heard of that in terms of, zebras running in the savannah or something like that. But a common pathway that occurs in humans and mice is this pupil dilation. So if you are freaked out or scared, or there's something really salient that happens, like an explosion next to you or something, you will have this immense response, and your pupils will dilate because of this stress response in order to get more information into your brain so that you can survive, essentially. And so this happens in humans. Humans, when they're resting, have a relatively normal pupil, but when they have something happen, it dilates. And this also happens in mice. Basically, what I'm trying to get across to you is that the brain receives information from the body, it interprets that information, and then uses pathways like this to send out signals to the body to tell it how to respond to certain inputs. So a good example I like to give is, besides this one, is the feeling of hunger, you know? So hunger, it's not like at 11:59 you're not hungry, and then at 12 p.m. you're hungry immediately. It's a gradual process, you know? It takes time to accumulate the feeling of hunger, and that's because signals that are coming from your bloodstream, such as leptin, or neural signals that are coming from your gut, which tell your brain, you know, this food was very nutritious, but you might want to crave a little bit more sugar, or you might want to crave a little bit more salt. All of that information takes time to be integrated in the brain, and then the brain needs to tell the body to engage either physiology like digestion, or a behavior like food seeking. Like if your brain were to detect you're hungry, it will make it harder for you to fall asleep, for instance, so that you can wake up, go and find food. So all of these processes are linked, is what I'm trying to say. And the last thing I'll get across is that, what does this have to do with cancer? Leah mentioned that we are a cancer neuroscience lab. You can't really understand the brain-body interactions in cancer without appreciating the normal brain-body interactions, so that's why I focused on that today. But in our lab, here's a very simple experiment that we did, and I'm sorry I'm showing you some nasty mouse tumors here, but these are breast tumors from mice that we've given breast cancer. And what we did is ablate the neurons, so the connections that the brain is making with the tumor. So we get rid of the input from the brain using a drug called RTX. And so when we do that, before we give the mice cancer, there's a couple mice that grow tumors. So there were 2 mice in this group that didn't grow any tumors. And then if we give it afterwards, we also get several mice that don't grow any tumors, and the ones that do grow tumors are much smaller. So this is something that we're very much interested in, is how can we interrupt this communication pathway between cancer and the brain to interrupt its growth, because what cancer loves to do is steal nutrients from nerves, from blood vessels, and if we can block that, I think this is a really nice way to complement some of the other strategies that are currently used, because there's no strategies right now that target the nervous system as an anti-cancer therapy, except for one that's in clinical trial in glioblastoma. But and then the last thing is, this isn't just something that happens in my lab. This is something that's been replicated many times. This is a study from 2017 where they show if you delete this one little receptor on the tumor cells, and this is the norepinephrine receptor, the thing that I showed you earlier, which drives the fight or flight response. If you get rid of that one receptor on the tumor cells, they don't grow at all. So in this case, this is a prostate cancer model, and you can see, just don't pay attention to this over here, but the growth curve here, you can see in the ones that have this receptor, the tumors grow huge, but in the ones that lack this, they don't grow at all. So basically we're trying to connect, how does the brain control things like this? What are the pathways by which it does that? And how can we block those pathways? So with that, I'll thank the lab. Honestly, couldn't have done anything without the lab. This is already going to be out of date next month, as we have 2 more people joining. So with that, I'm happy to take questions. Thank you.